So we talked about overrepresentation analysis. There is a second way to define gene sets or pathways that are affected in your data set. And this second way is called gene set enrichment analysis. And now we're going to talk about this. So in the overrepresentation analysis, we compared the list of up or down genes to the background. And the background was a list of all the genes that were tested for differential expression. The gene set enrichment analysis will work differently. So you don't start from your list of differentially expressed genes in this case, you simply work on the full data set. So the full data set being all the genes that were tested for differential expression. So basically this corresponds to the background in the overrepresentation analysis. So what are you going to do with these genes? You are going to sort them. You're going to sort them based on p-value. So the p-value that was obtained in the statistical test that determined whether the gene was differentially expressed, yes or no, or you're going to sort them based on log-fold change or absolute value of log-fold change. And log-fold changes are the log2 of the ratio of the expression in the treated samples over the expression in the control samples. So when you sort the genes like this, you get a ranking, and the genes with the lowest ranks, so the ones that are here at the left, these are the genes that are the most differentially expressed, while the genes with the highest ranks, that are at the right here of the ranking, these are the genes with the least differential expression. So in this ranking, you're going to look at gene sets. So gene sets are groups of genes that have something functionally in common. For instance, they all belong to the same pathway. So you're going to take all the genes that belong to the same pathway and you're going to look where they are in the ranking. So, for instance, for the red pathway, you see that most of the genes here are in the top of the ranking. So among the most differentially expressed genes. So this means that this pathway is very likely to be affected by the treatment that you have performed on your samples. Let's take a look at the purple pathway. You see that for the purple pathway, all the genes are among the top ranking genes. So again, you conclude that this pathway is affected by the treatment that you did on your samples. Now let's take a look at the blue pathway here. When you look at the genes of the blue pathway, you see that they are spread out over the ranking. So for this pathway, he will probably say, hey, this pathway is not affected in your experiment. Let's take a look at the green pathway here. So you see that the genes from the green pathway, they belong to the genes that are not differentially expressed. So here he will say, well, definitely this pathway is not affected by the treatment that you have done.